In GMP pharmaceutical manufacturing, isolators are essential, but maintaining these environments requires dependable, validated, high-level biodecontamination. As Annex 1 places greater emphasis on isolators and restricted barrier systems, the need for reliable, automated, compliance-ready decontamination has never been higher. This is where CURIS hybrid hydrogen peroxide and rapid vapor technology excel. For years, the industry relied on high-concentration vaporized hydrogen peroxide to keep isolators safe. But today, a safer, faster, and effective alternative has become the new standard: low-concentration mobile hybrid hydrogen peroxide, HHP, and integrated rapid vapor biodecontamination. To further understand the need for high-level decontamination, it's important to understand these 5 types of isolators, how they're used, and how CURIS low-concentration systems assist with contamination control. Containment isolators. Used for hazardous or potent materials, these negative pressure isolators require dependable closed-loop decontamination. Integrated CURIS rapid vapor systems provide faster cycles and safer material compatibility compared to high-concentration VPHP. Aseptic isolators. Critical for sterile compounding, gene therapy, cell therapy, and sterility testing. CURIS rapid vapor technology effectively treats both the isolator and its filtration system, helping eliminate potential contaminants trapped in filters. Flexible film isolators. Common in gnotobiotics, animal research, and healthcare. CURIS mobile biodecontamination units treat filters, transfer ports, and internal surfaces without the yellowing, damage, or multi-day downtime associated with chlorine dioxide or 35 to 59% VPHP. CURIS technology reduces full decontamination from days to under an hour. Rigid isolators. Used for cytotoxic chemicals and pathogen research. CURIS rapid vapor technology supports closed-system injection and automated extraction, providing efficient, contained, stainless steel-safe decontamination. Transfer isolators. Designed to protect materials during transfer. Traditional methods like peracetic acid or formaldehyde take days. CURIS Trinity reduces that turnaround from 2 days to just 2 hours with a validated 6-log result and no residue. The CURIS Trinity system brings all of this together in a mobile automated platform built specifically for isolator biodecontamination. CURIS hybrid hydrogen peroxide technology delivers 1. A safer 7% hydrogen peroxide solution versus 35 to 59% VPHP. 2. Automated, repeatable 6-log sporicidal efficacy. 3. Better material compatibility. 4. No corrosive or sticky residues, just water and oxygen. 5. Ability to treat isolators, filters, and contents in about 1 hour. 6. And low potential for staph exposure, enabling treatment even while rooms remain in use. Whether you're managing containment, aseptic, flexible film, rigid, or transfer isolators, the industry is shifting towards safer, faster, and more reliable decontamination technologies. CURIS hybrid hydrogen peroxide technology delivers the validated results, reduced cycle times, and Annex 1 aligned performance needed to protect your facility's sterility assurance.